Other formats that we've had over the course of this conference, we're going to have a series of talks from founders and entrepreneurs to discuss their experience and the lessons they've learned in bringing scientific breakthroughs to the market. So it's going to be a rapid series of talks. So we only have time for one question after each of the talks. So, just shoot those hands up and hopefully we'll be able to get you the microphone. So first, I would like to introduce our first speaker, Dr. Jake Redder, co-founder, director, and CEO of Celdara Medical.
Thank you, Elise. Let's see how that works. Wonderfully. Great. And let's see how this works. All right. We'll use this. All right. So my name's Jake Rader, and as mentioned, I'm currently the co-founder and CEO of a company called Celdara Medical. I'm not going to spend a lot of time talking about Celdara. It's a biotech incubator accelerator located about 2 hours, or headquartered about 2 hours from here. We do have offices in Boston as well as New York and DC. And we bring things from the academic stage through to first in clinic. What I did want to talk to you about was over the last 10.5 years that I've been running this company were some of the lessons learned that are applicable both for all the entrepreneurs in the room, I know there are many, but also for those people who are generally interested in innovation. I think these lessons are at once applicable to careers and to small companies. They're also applicable to individuals. And so the last thing I'll say about each one of these examples is that they're all a little bit non-intuitive. So hopefully you'll take something away that you weren't expecting to learn. There's some Latin up here. It's one of my favorite phrases. It means essentially that where you end up depends on where you start. And it's true and it's not true, right? I mean, all of us in this room started in different places. We've all ended up here. But what you learn on your path from where you begin to where you end gives you perspective, it gives you insight, and it gives you a lot of ability to increase the probabilities of success in your entrepreneurial journeys. And I'll highlight a few of those aspects as I go through the talk. So one other thing I'll say about beginning. When I was in grad school, I thought, I'm going to defend, and then I'm going to start. And what I realized very soon thereafter was that I had started 25 years earlier, right? It's a milestone, it's a nice thing, but it's a beginning of a career, it's not really a beginning. So let's start with the next slide here. Just a little bit of introduction so that you know what my own path looked like. I was one of these people who really never had a question. I always wanted to be a chemist. There was no doubt in my mind from the youngest age. Until about halfway through my PhD. At that point, I said, I love science, I love chemistry, but I don't have the patience to do the rigorous, high-quality science that is required. So are there other ways that I can participate in science and in entrepreneurship and innovation, rather, without having an idea and then spending 10 years trying to disprove it? And so I went into investment. I worked as an investor for a while. Then I went to a big company and worked doing strategic alliances with small companies as well as with universities. I then went up to the medical school at Dartmouth, a couple hours from here. And within 2 weeks of starting a new ventures office there, it was clear that what we actually needed was something like Celdara Medical. And so it took a few months, but then we co-founded the company in 2008. And then these last bullets give a little bit of the human side. I'm a first-generation immigrant. I am married to someone with an equally demanding job, and like all males in North America, I believe our relationship is balanced, independent of the data. I have 3 kids, a dog, and not too much travel. And I put this up here not to say, oh, look, there's a human side to this guy, but rather to encourage you to not think of your career in isolation. Because it is not in isolation. Even back in the 1950s, it wasn't in isolation. The lines were clearer back then, but now your work and your personal life mix to a very, very great extent. And so if you are optimizing for one in the absence of others, you're not optimizing. What has all this done? It's given me some perspectives. And this is something that, again, I hadn't predicted when I was first starting out, but having the perspective of the person across the table from you, really understanding not just what their job is, but how they think, enables empathy. And empathy is good business. If you can understand what the people around you are truly concerned about, what they get excited about, how they succeed, it's going to allow you to succeed more frequently. The good news about all of this is that you don't need to spend a career as an investor to get a very good sense and to get the perspective of an investor. A few years will do. You already come into this room with many valuable and diverse perspectives, and the question is, along your path, what other perspectives would you like to add in order to maximize your chances of success? So, how many people here have a 5-year plan? That's almost everybody. Good. It should be next GapSummit. That should be one of the qualifying questions. Good. Clearly, it means something. And that's great. 5-year plans are great. But I think you would do better if you started with a 50-year plan. What do I mean by that? One of the conversations that I have most frequently with postdocs and grad students and people starting out on entrepreneurial journeys is around what they actually want to do. So, well, I want to start a company. No, you don't. What do you want to do with the company? What do you want to do with your institution? What do you want to do inside the large organization? And what that question means is, what impact do you want to have on society? And that impact won't be accomplished in 5 years, especially in biotech. It's going to take longer. And so if you're projecting out 50 years, of course you will be wrong. You won't get it exactly right. But you will, through that introspection, get some insights into the types of things that motivate you and drive you, and that's going to set you along a better path than you otherwise would have been on. Another thing I recommend people do is find 10 people who are doing the thing they want to do, look at their biographies, find the patterns, find the trends. Going through that activity in a very tangible way will teach you something. So let's imagine you wanted to be the CEO of a large corporation. Time matters as an entrepreneur or an innovator. Risk always matters. It feels like graduate school has some risk associated, but really by comparison, it's quite low risk. But it's never zero, of course. There's always some risk in everything. And so if you wanted to march along the standard path or a normal path, you could go into corporate, eventually working your way up to CEO over many years. But this is America, and so in 60 minutes, 6-0, the state of not New Hampshire, the state of Delaware can form a company for you in 60 minutes. And you go to their website, and you could be the CEO within an hour. And then you can grow, and then maybe you go bankrupt, but your risk is going down as you're marching along here because you're learning, right? And so that's another extreme path you could take. There are lots of variations on this theme that are more common than the extremes, of course. And what I've found in seeing people who do this is that normal people, they don't love risk. They want to sort of stay away from risk. And the people that you see that are jumping in with both feet and going out and starting companies, typically no dependents, a trust fund, a lot of support, which in reality reduces their risk. Right? And so this personal risk and how you map out your path to whatever this goal is. God forbid you all become corporate CEOs. But whatever your goal is, map out the path and see how it looks from a risk perspective. Another thing that we often hear around, especially in this country, is that the government is a problem. It's bureaucratic. It's certainly not innovative. But let me show you some data. So this is the R&D spending of the top 20 pharma companies. The big wedge there, that's NIH. And so NIH dwarfs them all. And you say, well, that's, it's impressive, it's interesting. But innovation doesn't come from total R&D budgets. It generally comes from preclinical R&D budgets. And that's what preclinical R&D looks like. Now, it's not the top 20 pharma companies. This is all of them, every single one. Again, if you're looking for biomedical innovation, the government's funding it, and it's, this is the US only. Of course, most industrialized countries have something similar funding biotech innovation. And so does this really bear fruit? Do we really get more innovation out of it? And this is looking at cancer immunotherapy and the number of granted patents that come out of the university systems versus a per dollar normalized representative pharma group. And you can see that the universities are indeed producing more innovation. And of course, the governments are supporting those universities. Running a little short on time, so I'll say one other thing and try to upset somebody in the audience. Strategy is limiting. I used the original slide. I should have kept it. It said strategy is bad, but it's not always bad. Of course, strategy is a useful thing sometimes, but if you are doing something entrepreneurial, if you are doing something in a small organization, your primary advantage over the big guys is agility. Okay, so let's start with some definitions. This definition is always true, which is why I like it. There are hundreds of definitions for strategy, but this one's always true. It's a decision made at some point in the past to avoid certain otherwise perfectly acceptable options in the future. And that is always, whenever you see a strategy statement, you can say, okay, they're avoiding everything that's not on that page, right? And that's fine. If you've got a 100,000 person company, you need that. You need all those people to understand what the goals are and to be able to row in one direction. But if you've got a 5-person company or a 10 or a 50-person company, you can be agile. You can change direction quickly. And so optionality is what I would suggest for those of you who are developing innovation, who are concerned with being agile. You take your time, and instead of saying, these are the things I won't do, you build up your options. You can call these Plan B, Plan C, Plan D, for the times that things don't go as you hope, but they go as you expect. You expect that if you're doing early stage innovation, things will fail. And so expect to have to pivot, to have to move on to Plan B. And when you move to Plan B, now you better have a Plan C and a Plan D. All right, let me stop there. I'm getting the sign and see if there are any question singular.
Awesome, so we have time for one question. So who's it gonna be? The lucky one? No takers. There we go.
It is. So, hi, thanks so much for being here. My name is Zoe. I'm currently at MIT and Amgen. And one of the things I'm really curious about is how you actually marry those two worlds of strategy and optionality. Yeah. If you're a larger corporation that's trying to encourage innovation, and there are lots of different models to do that, sequester kind of teams or divisions away or do other things. I'm curious how you see those two worlds coming together.
You've actually mentioned the way that is most successful that I've seen is that you separate them entirely. And learning an operating culture from a large organization can be very valuable, but being defined by it or controlled by it or even reporting through it can be stifling. And so having the protected skunk works or phantom works or whatever you want and putting it to the side and giving it a lot of autonomy is one way. If you're thinking about, I was thinking you might have been going in a slightly different direction with the question, but if you're thinking about the growth of an organization over time, these are the growing pains, right? This is when you change from being able to be super agile all the time to saying, okay, guess what? We're making an investment, we're gonna go in this direction. And so I think of it sort of as a slider, it's a spectrum. Even when you're starting, of course, you've got some strategy, but you want more optionality. As you get to a larger organization, the options must become fewer because your focus and your guidance to your colleagues has to become stronger. All right.
Thank you so much. Our next talk will be by Dr. Christian Reichen, founder and chief scientific officer of Cristal Therapeutics.
Yeah, first of all, good afternoon, everybody, and thanks the organizers for being capable of presenting the main lessons that I actually learned when founding Cristal Therapeutics. It doesn't seem to work. So, well, what I like to give as a main lesson learned is actually about hiring the best experts worldwide. And maybe first giving you a little bit of a snapshot what CRISPR Therapeutics is all about. So we are a clinical stage pharmaceutical company. We're developing innovative nanomedicines. And I founded the company back in 2011, back at Utrecht University in the Netherlands. Why did I actually do that? Well, it was actually my PhD work that I was capable of actually bringing to the company. And the underlying reason why I really want to go forward is I've developed this technology, but clearly I want to see the true impact on the patient's field. And well, I had a little bit of an adventurous mindset there. What was initially the aim, kind of the dream? The dream is to bring drugs more selectively to the tumor tissue by getting them into the nanoparticulate systems, which are then accumulating in selectively the tumor tissue, so you really have locally the effect. While at the healthy tissue, the nanoparticles will not extravasate, so it will not be exposed, so you have a better safety profile. That was the dream. But then the dream actually in the last years came practice, since we currently have run 4 clinical trials, and we actually in the clinical setting demonstrate this improved safety profile. We demonstrate we have much higher uptake of the drug in the tumor tissue as a result of a much better biodistribution profile. And clearly also, equally importantly, we've evaluated already the efficacy, but clinical phase 1 trials are not really dedicated to really prove the efficacy as such, but we saw some promising evidence there. And well, at the moment, we're working to bring this product all the way to the market. At least that's the dream that we continuously still to have. And the actual application there would be in prostate cancer. So the aim is to go there for application of the phase 3 trial next year. And clearly, we want to do that on a global scale. So we're going to go for FDA advice, EMA as well as also aiming for Chinese input there. This is the company dreams. But clearly, the main question that I received from the organizers, what is the main lesson that I learned as a founder there? And well, maybe in the beginning I was a little bit of naive, I would say. That's really the first lesson learned, since the path to success is not as a straight line as is indicated here. I mean, in daily practice, an R&D project is really kind of complicated. And actually a lot of the projects fails because the science is hard, the biology is really complex, and we actually do not know that much as what we think we know. But clearly, the fact that I've done my PhD in this area is really very supportive, but you need much more to actually run a startup company successfully, much more competences there. And maybe talk about the financing part there. So you know the content part is okay, but you still need to finance the activities to go forward there. So we, as a company, in last years, we raised 20 million euros venture capital, as well as roughly 10 million non-dilutive funding, so governmental grants and so on. And if you then plot the overall timeline here, and clearly I'm not going to go into all the different details, but what I'd like to show you here is that as of the initial founding up till now, that the actual, every year almost there was a financing round or some activity there. So the financing part was really a core activity for the company. And that means that also, if you take a look at the numbers, we just had one of the investors of the Boston region. Clearly, the numbers in Europe are significantly less than in the US, but that's a whole different topic. I wouldn't address it today. So it has been a core activity for me to be in the financing part. And why was that? Well, to be honest, that was the only reason, not reason, but the only way we could actually finance the development. So we do had a couple of patents back in the days. We had some initial preclinical proof of concept, but there's nobody who was willing to actually already collaborate and pay and actually finance the overall development. So we had to go to investors to get this high-risk development being financed. Clearly, they aim for going for an exit with a good return on investment within a reasonable time frame. And that return on investment is highly influenced by the market as well as the competitors. And what I've noticed myself, and clearly this is my own interpretation here, is that VCs are highly interested to invest in companies once they have 2 critical elements, which is a very good business plan that really has a long-term strategy to make sure that the ideas that you have now are also still making sense in the 5 to 8 years' time when you have actually executed on them. As well, that you clearly be very transparent about all assumptions, as well that you have the plans validated by experts in the market. And the other key thing is the team. So maybe you have brilliant science, but if you do not have trust in the team, then the VC will never step into that. So you need to really have an established trust relationship, but also continuously building on that trust relationship. And that means in daily practice that you have frequent board meetings where you have to report on either the content progress, the timeline progress, as well as on the budget progress. And where they also really are eager that we are focusing on our core competences and not trying to invent the wheel on different areas ourselves. So to make sure that we really have the best expertise worldwide. And that's, as I said in the beginning, my main lesson learned is to really get the best expert worldwide for the advice or the execution of all kinds of topics instead of doing those developments ourselves in-house. And let me be very clear, this is about advising and executing. The final responsibility always stays at the company. So it doesn't mean that we simply give everything to the other party. No, it goes clearly definition of the aim, clearly being good communication, asking the really critical questions, what can an external party contribute as well as what they cannot contribute. And they can then clearly come to an agreement with very nicely defined success criteria before going forward. And let me give you a couple of examples there, what I found in recent years very helpful. So I said back in the days, we had only a couple of patents there, and one of the investors then asked, well, do you actually have freedom to operate? Then we did the analysis there. Probably you're aware freedom to operate means are you not infringing any other patents, so that would mean you need to have a license on that. And in fact, it turned out in 2015, we were infringing one of the patents. We talked to the parties that had those patents, but it was really difficult to get a license on that. And so overall, our assets, so one of the single assets we had, the value of that significantly dropped. So we need to do something there. And in consultation with a couple of both patent authorities as well as very smart chemists, we were capable of actually developing a complete novel class of chemistry compounds. So last year, we were very fortunate to both file a new patent, which is good because we have an additional asset on our own hand, as well as we now have also freedom to operate. So that was very supportive for us. Another thing is, well, you can have nice data, but you ultimately, if you want to bring it to the patient setting, you need to produce it under GMP conditions. Well, production, I produced the material myself back in my PhD work, so this is literally a picture of the material produced. So the opalescence really illustrates there are nanoparticles in there for the experts in the field. So I was capable of producing the material at a small scale, 1 to 3 milliliter scale, but clearly you need much more for the clinical application, and it needs to be produced under GMP conditions. So already back in the early days, 2012, we collaborated with an experienced CMO who all the way upscaled to 40 liters, made it fully compliant to set GMP criteria, and made sure that we had, and this is just a showcase of the certificate of analysis, we had material clinical batches being produced fully compliant to the set criteria, and meaning that also we had always sufficient material for the clinical trials that we were running. So the two last examples are about companies that have so much expertise. They've done this many, many years, so they do have a lot of expertise there, but it clearly goes against the price, so it's quite costly to do all those developments. And to be honest, I would say that the real innovative developments, they're not done at these companies, but rather they're done at the universities. So when we had the idea to actually go for the nanoparticles and go all the way for non-invasive imaging, so that means that we simply needed to conjugate a PET label to the surface. Then we went to actually the VMC at the Amsterdam University Center because they were worldwide experts on getting these type of zirconium labels to the nanoparticle. And without going into all the details, we now have been capable of showing actually that this is where you take a look at the zirconium labels, showing in the clinical setting that we do have accumulation in the tumor tissue. These are very brand new data since, if you ask me honestly, how long this overall development took, the overall idea was, the initial idea was in 2007. These pictures are just out of March, so it took us more than 12 years to actually get the overall development done, meaning you need to have the best expert there and to also be clearly a little bit persistent in this area. Well, so far I've talked about mainly the content, but clearly you can start up a company by yourself, and then you have this pioneer environment. But that's a whole different environment if you are now what we currently are, a company with clinical candidates. We have quite some collaboration with big pharma as well as biotech companies. So then once the co-founder, he retired last November. When he retired, then we said, okay, well, this is also the opportunity to go for somebody who has really also the background to bring the company to the next level. So the co-founder was really pioneering, entrepreneurial, was capable of raising money because of his pioneering approach. But currently, we need somebody who's a more, I would just say, it's a bigger company, more structured, procedural approach. Maybe I have to say the company is quite small still. We're only 15 people, so 1-5. So that's, I would say, quite small. But what we've done also in recent years is really spend some additional time on training the team. So meaning that identifying leadership potential, making sure giving those people more responsibility so they can further grow, as well as also provided additional communication training so to really have good interaction amongst all the different people, both internally as well as with all the external experts, as said. And with having said that, I think to make a long story short, if you have a good plan and a good team with a lot of experience that should be capable of raising sufficient venture capital financing, then it's a matter of only executing as you agreed upon, including clearly the risk mitigation approach, whilst assuring that you have a good team spirit. That in combination with the external experts, I think that should be enough success factors to have a straight line towards success. And with having said that, I'm very pleased to answer any questions now or maybe later on.
Hi, Luca from EasyCarp here, an early stage digital health startup from Slovenia. So you mentioned both VC funding and grant funding, which you got quite a lot of it. My question would be, so grant funding is pretty compelling for early stage startups because it usually doesn't take any equity. But do you think that it's a worthy investment to work on all those grant applications because they take a lot of time, especially for early stage startups, or is it a better idea to go after accelerators and private funding? Thank you.
It's a very relevant question, so it's kind of the balancing part. But I would say for Crystal, the actual governmental subsidies, grants, they have been very instrumental in actually getting the company launched. So in the initial years, we overall get 1 million in the initial first years to really get the proof of concept, and that really helped us going to VCs saying, okay, we do not only have a concept, but we have data generated, and that really opened the doors to the VCs. So these days, we are indeed lesser interested into going for example, the bigger grant consortia, where we can only contribute to a minority, and we are not really in the driver's seat dictating where the program should go. But we are only interested then to join if it's really something that we're eager to learn, but not really on the critical path ourselves. So these days, we'll be more focused in going for venture capital money, indeed.
Probably have time for one more question.
Hello, I'm Vicente from Spain, and I would really like to hear more about the role of the university in this kind of a spin-off creation. Was it like a booster, or was more like a hindrance to the creation of the company?
Yeah, so when I started my PhD, the project was already launched, and actually there was already one patent filed on that. And actually during the first 2 weeks of my PhD period, I contributed, so I became a co-inventor on the patent, meaning that my supervisor already had kind of a feeling that all the developments, if feasible, need to be patent protected. And on the long run, the actual university and also the TTO correlated to that, they were very supportive in getting the initial findings actually brought towards further stage development. So they were was a very constructive support. And even these days, the Utrecht University Holding, which is a small investment fund, they're also actively contributing to bring the company going forward. So they're actually investing at the moment.
Thank you so much.
You're welcome.
All right, next speaker is Dr. Eric Olenko, co-founder and chief innovation officer at PureTech Health.
Great. It's great to be here and to tell you about PureTech and the way we go about and do innovation and create medicines. PureTech is, oh, no problem. This is a short disclaimer from our lawyers, by the way, about forward-looking statements. PureTech is focused on creating therapies for disorders of the brain, immune system, and gut. And we're particularly interested in the intersection between these three systems, or what we call the big axis. Now, the way we create these therapies is different than the way that most people go about innovation. Most people start with a technology, a breakthrough, and say, how do I apply this? We do the opposite. We start with a problem. How do we have a safe and effective treatment for obesity? How do we have a new first-in-class mechanism and way of treating schizophrenia? How do we treat autoimmune disease so we only treat the disease and target the disease, but we don't affect the rest of the immune system? Now, what we do is, once we have this problem, we'll go and form a group of leading people in the field and start to brainstorm what potential solutions are. From those brainstormings, that defines a boundary of a solution set that we then use to go and proactively look around the world for technologies that could be a starting point for solving the problem. Once we find technologies, we go and then we put on our skeptic's hat. We say, well, what are we most worried about? Why do we think this won't work? And then we go do the experiments that'll show either, yes, this could be a real solution for this problem, or no, it's not. If it's not, we do the experiments and we kill it. If it's good, it moves on, and we further develop it. And it goes into one of our two divisions. We have an internal division, which consists of programs entirely owned by PureTech. And then we also have our affiliate division. With our affiliates, what we do is we start new companies, put the technologies in those companies, fund them, and drive them forward. And then at the appropriate point in that affiliate's maturity, it'll go out and seek third-party capitalization. Now, what I want to do is give a brief case study about one of our affiliates called Genesis to give you a sense of how this practically works in terms of a problem-based approach. We were really interested in tackling the problem of obesity and people being overweight, arguably the largest healthcare problem that exists in the United States, and many problems around the world, and it's been quite challenging. When we got together leading experts and we brainstormed, everyone converged on a certain set of criteria for the ideal product. It would be something that would be safe and well tolerated so that people who were at a lower body mass index or BMI, people who were overweight, not just severely obese, could use the product. It would be one that would be taken orally, and it would actually be one that would act mechanically, not pharmacologically, to induce multiple mechanisms of satiety. So based on this criteria, we went, did a proactive search, and one of the technologies that we identified, which became the lead program for Genesis, we went and took forward. And this consists of small particles, which are packed in capsules, which somebody takes before a meal. Those particles then absorb water and expand in size about 100 times in the GI tract, inducing a sense of satiety so someone eats less. They're engineered so that in the large intestine, they break down, releasing the absorbed liquid, and then pass out of the body in the same manner as food. These particles, they're proprietary in nature, but the building blocks are actually naturally occurring, as you see on this slide. So this was an example of finding a solution that was really out of the box and led us to really lead the way in this new area of mechanotherapeutics using a mechanical way of inducing a therapeutic effect. I'm going to fast forward a couple years in development. This became the product name you see there called Plenity. You're seeing the results on this slide of a pivotal trial that was conducted with Plenity. A statistically significant difference from placebo. As you can see, at 6 months, 59% of people lost 5% or more of their body weight, which is what's considered medically meaningful. And a greater percentage of those, there was a certain percentage of individuals who actually lost an even greater amount of their body weight. This was accompanied by an excellent safety profile and tolerability profile, which we had identified early on being really key in terms of working in this area. And this resulted earlier in the year in actual FDA clearance of this product, which you can see here, the potential packaging and what this could look like. So this is an example where we went from literally a blank sheet of paper saying what's the ideal solution to a problem to actually going out to the world, finding something, building it up, and this is through our affiliate Gelisys, and then actually getting something FDA cleared. So just wanted to give that as a case study. Now, we think this kind of problem and Gelisys, by the way, has a whole platform of products that it's working on for a number of other uses outside of obesity. We think this is one example where you can use this problem-solving based approach. What you see is our various affiliates. We also have our internal pipeline. And what each of these various affiliates have in common is that they're each taking a real first-in-class approach, or in certain cases, they're actually defining entirely new areas of medicine. So in the case of Gelisys, mechanotherapeutics. In other cases, such as Achille and Son, digital health, you can go down the list. But by starting with the problem, we can really start to think out of the box and identify solutions that might be somewhat more unconventional. And I'm just going to end the talk, since I know there are a lot of younger entrepreneurs in this audience, with this slide that, our experience is when you're looking at something new, of course, you have to put on your skeptic hat and ask, what don't you believe? Do the right experiments to kill something, and do things based on empirical evidence. But in terms of the skepticism of, well, no one's done it before, so it can't be done, I would say that's just not true, since clearly people have shown that it's possible. So I would really encourage people that if you do have that idea you believe in, even if it is something out of the box and hasn't been done, that you should pursue it, and it's definitely something that's possible. So I will end the talk there, and happy to take one or two brief questions about PureTech or the case example that I gave.
Thank you. Very interesting model on how you scale for technology. I'm Giacomo. I'm a biotech entrepreneur. I was wondering, do you use specific methods or techniques to scout around for exactly what you want, patents or publication, key opinion leaders? If you can share, if it's not confidential. Thanks.
No. I think so. What we found that's really key is in that beginning process, when we're getting people together and we're brainstorming and we're saying, what's the ideal solution? When you get that right set of people together who are really the key leaders in their field, and it's an interdisciplinary group, they really start to point where to look. So it's a combination of, and by the way we tend to look is to go right to the scientists. So most of what we have worked on has been something that's come out of academia. We have had things that have come out of industry. But what we find is they'll say, well, so-and-so is doing something really interesting, and it meets the criteria that we talked about in this group. Go speak with them. Or, so-and-so, I know they're about to come out with a paper, or I was just in a conference, go speak with them. So we found that human element is very important. Of course, we'll do things like look at the literature, look at various, the IP that's out there. But we found it's really that networking effect. Where we found the best things where we have kind of asymmetric knowledge of, well, we know that this is something that if we could do, it'll solve the problem. We know this person actually has something. Now let's put them together. And that's how we end up with these kind of first-in-class unconventional approaches. Is there another question?
Hi, my name is Daniela, bioprocess engineer. Actually, my question was super related to Giacomo, but now I opened up the space for another one. So I'm really interested in how do you actually got the funds to create PureTech, since it's more of a networking effect or totally different approach of the companies we have seen today. Thank you.
Sure. So PureTech itself is a public company trading on the main market of the London Stock Exchange, which is why there was that lengthy disclaimer in the beginning. So, PureTech and I really have to credit our CEO and founder, Daphne Zohar, who really got this off the ground from scratch. And really, the process was building, getting the first money in from private investors, starting to prove out the model, bringing in some programs, showing that an added value, and then kind of building up over time, which eventually, resulted in us having the capital that we have and being able to go public on the exchange that we did. So it was a process, over time, but I think it was kind of a combination of having the right people, having that credibility, starting to build it out. And again, in the very beginning, I really have to give credit to Daphne for making that happen really from scratch.
Great, thank you so much.
Great.
Next, I would like to introduce Dr. Jason Kelly, co-founder and chief executive officer of Ginkgo Bioworks.
Awesome. Okay, yeah. Cool. Thanks, everybody. So just to give a little background on Ginkgo, we're about a 240-person company located over in the Seaport here in Boston, about 10 minutes away. Raised about $430 million, last round at over $1 billion valuation, and we're a hard tech company, so I'll give a little bit of background on what we do, but the founding team was a group who met in grad school, and we started the company out of grad school, and I think that's a phenomenon that in the world of software, you see a lot of companies get started with students out of school and then grow pretty big quickly. It's more rare in hard tech, so I'll try to share why I think it's hard, why there's less of it, but that's not insurmountable, and some of what we learned in doing it. But I'll give you a quick minute on Ginkgo just so you have the context. Essentially what we do, this is an article I like, Wire Magazine, move over Johnny Ive, biologists are the next rock star designers. Basic idea behind Ginkgo is we want to design biology, right? And we want to do that by engineering the genomes of cells, and we want to do that in a generic way across many markets. And many of you in the room are sort of biomedical engineers, but the basic idea behind Ginkgo is DNA is code. We should treat it that way and program it. We're the largest user of DNA printing in the world. By our estimate, about 25% of worldwide DNA printing is done by Ginkgo, ordered or manufactured in-house. We acquired a company called Gen9, a local company here, about 3 years ago to bring that in. And then the other technology we have is basically automating the work that I did during grad school, which is a scientist at the lab bench doing protocols to genetically engineer cells. We have about a 100,000 square foot facility today. It looks a lot like this. Lots of robotics and automation to bring down the cost of doing that work. So for about the last 5 years running, we've been 3xing our output in the facility and dropping costs by about 50% per year. That's a very traditional scale economic that you see in things like auto manufacturing or electronics manufacturing. We believe it's fundamentally new in the area of cell programming, cell engineering. We then have applied that across a range of markets. I think you'll actually hear from Bob tomorrow from Bayer, but one of our projects is to genetically engineer the microbiome of crops in order to do things like produce fertilizers. This is to work on nitrogen fixation in corn. We've also done work in cannabis. We've done a $90 million project in animal proteins like milk without cows, eggs without chickens. We announced a deal with Roche in antibiotics discovery. We work in things like with the Defense Department a bit in biosecurity. About 50 projects today across 25 customers, again, operating as a platform to enable cells to be programmed for a wide range of applications. All right, so that's Ginkgo. Good, I got plenty of time left. All right, now let's talk about the important stuff. All right, so how did this happen? So the company actually got started initially in 2008. Bootstrapped, so the first money into the company was about $120,000 from one of the founders of the company. So there's 5 of us that started it. 4 of us were in grad school together here at MIT, over in the Stata Center and in Building 68. And the 5th founder was a professor at MIT, Tom Knight. Tom put in that $120K and agreed to be a founder. He also left MIT about 2.5 years later, so like a little bit of a thing to keep in mind when you're launching these companies. There's often sort of a professor founder that's kind of tagging along and taking 5 or 10% of the equity. That's not a good idea. And so, yeah. Hang on to that equity. But if they will leave the university or put some dollars up, those are good signals. Really, we didn't have any capital at the beginning, so did a lot of eBay, A lot of when labs would shut down at the university, it would work like first the email goes out to the department mailing list, hey, the lab's shutting down, does anybody want the glassware? Then it goes out to like the whole school, hey, the lab's shutting down, does anybody want this other stuff? And then finally the last email is sort of like, everything in this lab is going in the garbage at the end of the week. Literally anybody can take it for anything. And MIT's pretty good about that, a reuse list. And so we would, at that point, show up and sort of harvest all this junk. This is literally a sink, okay, right over here on the right. On the left, that's about 30-year-old fridge that we got for 15 bucks from some old lady in South Boston's basement to use as a lab fridge. All right, so just to give you a sense of, you can be cash conscious at the beginning. We got a $150,000 check from the city of Boston to move out to the Seaport area. This was part of Boston at the time was like derelict. Now it's pretty popular. But this is a lot of money for us at the time. And just to flag it, government grants got brought up earlier. Between 2008 and 2014, we probably applied for about 100 government grants. We really ran that almost like a sales pipeline, applying all the time. Probably about $15 million over that period. We were not venture fundable at that time. Number one, the life science industry is like VC community is not very good at funding people straight out of school. That's not like their worldview on what a founder looks like. I disagree with that, but it is what it is. And so I would not, if you are that phenotype, I would recommend going out to government agencies and try to get capital. And then, as mentioned earlier, we started taking our first, we really didn't take a customer until about 2013, initially in the fragrance industry. And once we had a couple customers, then we had a story I thought that was a little more amenable to a place like an incubator that got mentioned earlier. So we did Y Combinator, summer 2014. We're the first life science company they had backed. This is actually becoming much more popular now. So if you were going to try to raise your first, call it $1 to $5 million, I'd recommend just like go get an Airbnb in San Francisco and like hit up coffee shops, do an incubator, and like, a couple $1 million will fall off the back of a truck. It's going to be easier to get money that way than to like go to traditional venture again if you're like people right out of school. And then like I mentioned, across 3 rounds, about $430 million. Most recent round in 2017 with General Atlantic coming in for $275 million. And now the company getting mentioned alongside other fast-growing tech companies. This is CNBC's list of top 50 private companies. Biotech getting involved, hard tech getting in that same list as the software companies. Okay, so that's sort of like quick timeline on Ginkgo. So now I want to just talk about a couple structural issues that I think are you should worry about when it comes to hard technology. So I call this the SpaceX problem. So it's sort of holy crap, SpaceX like landed 2 rockets simultaneously. Like that is an amazing achievement. It's really great that we have that company. However, that company basically only exists because of like Elon Musk's like exceeding wealth and his ability to go take a flyer on it, right? It's not being produced rationally by our venture capital community. That wasn't like something that fell out of the economy in the like proper way we start companies. And so why is that? And my argument is that tough tech or hard tech or whatever you want to call this stuff that could be the basis for successful corporations is actually abundant. There's tons of SpaceXs lurking out there. And so why aren't they getting funded all the time? Why does it require some billionaire to go do this? And I think there's 2 possible limitations, either talent or capital, okay? And the core challenge is that doing like a rocket company, doing a biotech company, doing a high-end materials company, these all require very specialized knowledge, right? And so to speak to the talent question, I actually think this is not the issue at all. And the beauty of this is that the academic system is producing unbelievably niche, talented people that know exactly about one specific thing. And they also have some of the features that Paul Graham at Y Combinator, and if you read the link down here, this is a talk he gave at MIT like years ago in 2006 when they first started YC, where he was saying, hey, here's what makes for a great startup founder. Okay, and I'll argue this is exactly the same for PhDs, but he was talking about undergrads. He said you need to have stamina, so it helps that you're young and you can have all the energy to do this. It helps to be poor, okay, because you are going to have to eat ramen and push through it and not get paid a salary. And if you have a huge amount of expenses, that's not going to help you. It helps to be rootless. Like you need to be able to move easily. You might be able to have co-founders who need to move. It helps to have good, smart colleagues, right? And there he was like, listen, at the university, there's going to be other really great co-founders around you. And it helps to be ignorant of how difficult it's going to be to do this, okay, right? And I would argue that's exactly the same situation for any graduating PhD or postdoc, right? You certainly still have a lot of stamina. You are certainly poor. You are rootless. And by the way, you're looking for good colleagues. You know, my co-founder, Barry, you turn around, it's the person behind you at the bench, okay, right? You know, like the other people in your lab, and you are ignorant of how hard it's going to be, okay? I think the issue is capital, okay? And in fact, late stage capital is actually easy. So at the stage we're at now at Ginkgo, it's far easier to tap capital, right? Like, because you have evidence. You can point to products coming out. You can point to customers that are validating you. At that stage, it's actually quite easy to tap capital. The issue is the early stage, and not even like now I think at least in the current environment, the 1 to 3 million is pretty gettable because you can get it from just like angels who are like, woo, you know, right? Like that sort of thing. But it's that 5 to 30 million, all right, that's going to trip you up, right? That is the really difficult capital to get for hard technology companies. And there's a really rational reason for this, okay? Which is that if you're a venture capitalist and you're pursuing companies that come to you with business risk, right? Like, will grown adults ride scooters, right? That's business risk. Can I create a network of scooters that I can rent with my phone? Of course. There's zero technical risk in that, right? Whereas all the things you're talking about doing, they have deep technical or scientific risk. If I don't have the ability to evaluate that as the investor, how can I make a good decision prior to that technology being validated? That's the issue. And even if I'm a specialist, like I know about this one area, are there enough companies showing up with this one niche area of expertise I have? Rarely there is. In pharma is actually a unique exception where there's enough deal flow for that, but for the most part, no. So what could you do about it? All right, I'm gonna give you a few options. Number one, hustle, okay, right? So this is a bad graph over here on the left. This company is worth nothing until I get an amazing result after spending $25 million on all this technology development, and then I will present to you my genius, okay? That's really tough because that means you gotta raise $25 million when the company's worth nothing, you're gonna lose the whole company, all right? What you want is something like this, like, as the company becomes more valuable, okay, as I spend money, the company goes up, and I can raise anywhere along here, and it's an efficient place to raise money, right? This is like what Facebook growing users looks like, right? So how do you do this if you have to get through like a phase 2 clinical trial or something, right? Well, the answer is you can tell stories, right? So this is a company from Y Combinator that is doing supersonic jets, okay? And what did they do during Y Combinator? Well, they cut a deal with Richard Branson. And that was enough press and PR, blah, blah, blah. They could raise 15 or 20 million bucks at a stepped up valuation. So even though they didn't have their jet working, they had spent some money and created value in the brand, right? So you can hustle, you can find ways to do that for hard technology. I think it's really important, it's a great solution. Number two, government. We did this is awesome. Go do it, go get the government money, it's non-dilutive. First 20 million into Ginkgo. Third, get government agencies to be that credibilizing thing. So the pharma industry has done this really well, where they say, okay, well, you don't have a drug out yet, but I have blessed you with success in a phase 1 trial, so your valuation goes up. Number 4, rely on the whims of billionaires. I do not find this to be a scalable solution. And I'll leave you on this. I think actually now is the time for hard technology. The world has really big problems. Nobody wants to work for like advertising companies anymore, right? I mean, it's just the tech industry is just soulless, right? And so like this is it. The types of things you're working on are really the future, and so you should embrace it. And good luck to all of you. Thanks a lot.
Question, start. Cool. Name's Sam. Set up an early stage biotech called Phenomic AI. Very, very similar stuff. Way small. Just on our first seed. We did a seed round last year. So question for you: which was like your hardest funding round, and why? And how did you get through that?
Yeah, I mean, so I would say, I mean, and remember, we were doing this, in 2014, but it was probably the round out of Y Combinator where we were trying to raise, call it $20 million, and that was like only going okay, and we basically I'd say we got lucky, like we got connected to a fund that came at us from another direction. We're interested in genomics, and we really fit their profile, and they went for it. But in the absence of that, that would have been the toughest round. And I think it is still, and now I think it's gotten a little better, but it's still that $20 million B or whatever you want to call it. That's the round that trips up most of the hard tech companies. Yeah.
Hi, Jason. I've actually followed your company since, I think, late 2009, and I even know the evolution on your website, funnily enough. So I'm really glad to hear from you. You mentioned that it took a while before you could start generating revenue, right? 2014 is what you were saying?
Yeah, it was 5, 6 years of
Yeah.
technology development. Yeah.
So how did you identify that first route to market, and like kind of how did you make the journey? Because that seems to be one of the biggest challenges.
Yeah, that's a really good question. So we were very, I mean, Ginkgo's, it depends on the technology you're building. This is very specific to us. But we adopted a model of being very opportunistic about where the technology could be applied. We had a strong vision for what the technology was. And then we were just like, test, sort of lean startup methodology, if you're familiar with that, just sort of march through different markets, and that was it. And then falling like a ton of bricks on anything that looked like it would give us a customer, because you just don't have any credibility, right? And so every one of those partnerships stacked credibility, and then we could build on that. Yeah.
Hey, Daniela here. Building onto that, like, how did you actually engage with your customers so that they will help you to validate your technology while protecting your IP and your value?
Yeah. So a couple things. One, we were extremely generous in the types of deals we cut initially. Because I felt that the technology fundamentally had a lot of applications. And so it was more important at the beginning, you know, if I could give a great deal to a fragrance customer, but it would validate that some company actually cared about my technology at all, that wasn't me, that would incrementally increase the value of the company much more than whatever I gave away in that project. And so in that beginning period of time, don't underestimate like how low the cachet of a brand, even today, nobody knows who the hell Ginkgo is, right? Like the amount of value we can still create through brand awareness is enormous, through credibility is enormous. So give it away, you know, right? Like unless your technology really is just one thing, then you have to be more careful. But like if you think it's something that has a variety of, you know, if it's a platform or has a variety of uses, then just be generous. That's what we did.
Great, thank you so much.
Yeah, thanks.
All right, our last speaker is Dr. Sangeeta Bhatia, a Howard Hughes Medical Institute investigator and the John J. Dorothy Wilson Professor at MIT's Institute of Medical Engineering and Science, Electrical Engineering, and Computer Science.
Hi. Oh, okay. I have to use the podium. Hi. Can you guys hear me? So I have to have this on and talk here. Oh, those don't work. Okay. Whatever. I'll stand here and then we'll figure it out. Oh, I see. The clicker doesn't work. Okay. All right. Okay. I'm going to tell you the opposite story of PureTech, which I love, which is the way that we invent things in our academic labs at MIT and get them out into the world. And so this is a talk that I've given different versions of, and it's sort of in a longer version, there's sort of like a pick and choose. You can actually ask me to tell you which story you want on the next slide. But the title of it is, so you've made an invention you think can help patients, now what? And when I first started making a list of inventions that had come out of our lab, this used to be the 10 tiny technologies talk. So my lab uses micro and nanotechnology for medical applications. And the list has grown over the years, so now it's 15, and this is an incomplete list. The red things have been things that turned into startups. The blue things are things that we're kind of brewing for future startups. And the black things are things that at some point the trainee in the lab thought about starting a company around, and there may be a way to get to patients, and it may or may not actually go through a company as a route to getting to a patient. So there's a lot of different stories up here. I wanted to tell you just 3 quick vignettes, and then sort of what the lessons and the themes are that came out of them. And in general, what you'll see about these 3 stories is that one theme is, the reason this is the opposite story from PureTech is that we actually usually are doing basic science in the lab. We're asking some fundamental question about the structure of a tissue or whether a particular material can enter into a disease state or make a measurement. And once we have that sort of capability, then we start to sort of turn it around and ask, like, is this useful clinically? What would the killer app be? We talk to some people, we change it some more, and we find our way to sort of what I call a clinical use case. So it's almost the opposite of starting with the end solution. But you know, we're tinkerers, and that's how we do things in our lab. And so this is the first one. This is like Christian's story. This was actually originally the foundation of my PhD thesis, and then when I became a professor, it became a subsequent PhD thesis. So it was 2 PhD theses. And the basic question that we were asking was, how can we stabilize liver cells outside of the body? And so we did that by microfabricating these plates, clustering them together, hepatocytes, on a surface and surrounding them with supportive cells. And my whole PhD, I just studied this, which neighbors do they like, how big should the colonies be, and the goal of my PhD thesis was supposed to be, once we had stabilized these, to build a big extracorporeal artificial liver device, so a cartridge that you would run blood through and for patients. That was the idea. And so I did that in rat cells, and my PhD student Salman Khatani then went on to do it in human cells. And we're very interested in saving patients eventually, but somewhere along the way we realized that actually we had a human liver in a dish, and that might, in and of itself, be a useful invention for the pharmaceutical industry, for predicting the safety and metabolism of drugs, which happens in the human liver in a way that can't be predicted in animals. And so we set out to start a company around that idea. We did that in 2008. We've been talking about financing models. We did that with venture backing. The 2 of us were co-founders with a third person, a business partner, a CEO, that we met in the MIT ecosystem that you've heard about. And so the way this works is Salman and I were thinking about doing this. We wrote a grant to the Deshpande Center, which is our local accelerator. And Lita Nelson, who was the head of the TLO at the time, started telling people in town, hey, Sangeeta's starting a company, go by her office. And eventually we bumped into our third co-founder, Bonnie Fendrock, and she started the company. And we raised this $5 million of venture capital. She had, I've forgotten whose slide had this on it, that you have more risk when you have a spouse who is well-employed. The first speaker had that. So she had a husband who still has the husband, who was a professor over at Harvard at the Dana-Farber Cancer Institute, and he had said, Bonnie, I'll give you a year. And so we had a year of Bonnie's time for so-called sweat equity, where we were going to go raise this round. And I had my second child in the crib, so she was a newborn, and they would come to my house every Tuesday night, and we would work on this business plan. By the end of the year, the 11th month of the year, we raised the round. So that became Heprogen. The technology's been scaled up. We got acquired last year. You can sort of buy it all over the world, and so that's one story. That's a tool story. The second story I wanted to tell you was this story. This is a story of a company called Glimpse, which we founded more recently. This was also, so this is a technology for non-invasive disease monitoring. You inject nanomaterials into the body. They sense enzymes that are disease-associated. When they get activated, they put a signal into the urine that you can measure remotely. And this technology was also not intended to be this in the beginning. So what we were trying to do was make materials, nanomaterials, that would enter into tumors and be MRI contrast agents. And the idea was that they would be smart MRI contrast agents, not unlike what Christian showed in the terms of the ability to visualize parts of the tumor. We were trying to make them responsive to the enzymes in tumors. And the idea was that they would cluster together in the presence of certain enzymes, and that would give you a signal on MR. So you could see like what part of the cancer was invading. That was the idea. By the way, it never worked. For all kinds of reasons. But the students who were doing the project noticed that whenever the animals had cancer, that the bladder was lighting up. And we had sort of this aha moment, which is, oh, we can inject these materials, we can detect a signal in the urine, and that became sort of the aha moment that we didn't need an MR machine at all, and maybe we had invented a diagnostic technology. So here again, we went to the Deshpande Center the second time. And here, I think we've been talking about different models of seed funding. It is true, I think, in this current moment that it's not difficult to get the first sort of $1 to $5 million seed round. So we were seed funded initially. We spun out. And then that second round, the VC-backed round, I actually took a sabbatical for a year to run the company at the advice of Bob Langer, who's a wonderful mentor, and raised that bigger second round, which was a $22 million venture-backed round. And so I spent all year, in Bob's words, giving it a push. and hired a professional team and came back to MIT in January, and that team is now taking it into first-in-human clinical trials next quarter. So that's the second vignette. So this is a second technology that's not how we thought it was going to be, and in a different sector completely. So the first one was tools. This one is diagnostics. My co-founder here was also my trainee, Gabe Kwong. And this is a whole different story, and it's just a story of multiple ways to do things where he actually was a postdoc and wanted to be a professor. So he co-founded the company with me, but he went off to be a professor, and the two of us started this company with actually like nobody in the room until a graduate student named Andrew Warren decided to help us be employee number one. So it was a sort of different cobblestone together kind of team. And I think you can build a team a lot of different ways. The last story I want to tell you about is a story about exactly what you heard about, which is the student next to you, the person in the next bench. So this is a company that came out of my lab. The technology was invented in my lab and morphed, and these are three friends in my lab that didn't start the company. I didn't start the company, and they didn't start the company when it was in the lab. And what happened was they actually graduated, went away, and all got jobs. And one of them, named Todd Harris, had this idea one day with his wife about his friend's PhD project. So not his project, his friend's PhD project. And that friend, you'll hear from him later, was Jeff von Maltzahn, who went on to become a VC. And Todd and Jeff were friends. And what Jeff had done was inject gold nanomaterials into animals and make them sensitive to near-infrared light to do cancer hyperthermia. And Todd is blonde, and his wife, Ashley, is blonde. And Ashley came home one day and said, I can't get laser hair removal because I'm blonde. And Todd remembered Jeff's nanomaterials and that they were sensitive to near-infrared light and had this aha moment where he thought, like, oh, if we put them on the skin, nanomaterials will go in the hair follicles, and if we shine near-infrared light on them, then we can sensitize Ashley's hair to near-infrared light, and that will be laser hair removal for gray or blonde hair. Todd at the time was actually at McKinsey, had a day job. They had 4 children together. So he called Jeff, his old friend, and Alice, a third friend from my lab, and together they co-founded Sienna, even though they all had jobs and were in 3 different cities. And they raised a small amount of angel capital. Eventually, they got this working through in this kind of bootstrappy way, and eventually Todd left, ran Sienna, got venture-backed, went public last year. So that's Sienna. So that's a third story. Again, totally different model. So many, many different ways to get from an invention to a patient. I would say, in my experience, things coming out of my lab never look in the end how they started. And so you have to constantly iterate and figure out kind of what you have. So this is for those of you who are interested in other inventions, please come see me later. These are the lessons. What do you do? So you have an invention, you want to get to patients, what do you do next? So I think the first thing is zip up your invention. And you've heard about the importance of publishing it in a peer-reviewed place, so zip it up, protect the IP. Third, make sure that you have not just the IP in the papers, but the know-how. So I think you've heard a lot about how the technology worked in somebody's hands and not someone else's hands, or it worked in that lab in another lab, but not once it got into the incubator. So make sure that as a team, you really have the technology working robustly. I can't emphasize that enough. The second thing is to clarify the path to patients. So focusing the use case is something that I think you can do as you go. So you kind of have an invention. We do this in our lab all the time because we just have clinician scientists around, and we're sort of always saying like, hey, is this interesting if we can do this? Or how do you do this? Or, oh yeah, we invented this in cancer, but it's really useful in the skin. Or yeah, we invented this for liver disease, but it's actually really more important in diabetes. Because you don't have to get attached to like where you thought the invention would be useful. It might be useful somewhere else in medicine. You should consider many different business models. I've only told you startup business models, but you can actually take things to patients purely through licensing. The TLO can help you with that. You can do an investigator-initiated clinical trial that's collaborative. You can do that with grant funding or institutional funding. So there are other paths to patients than just venture. And then I think a bunch of people have said this. You should be a dreamer. I think that if you know your technology, you're the only one who can imagine what it might be someday, but you have to be practical and opportunistic around the way. So there's this sort of constant battle of being like the visionary and the pragmatist. But you should still be the dreamer because you're the only one who's going to actually be able to dream what your technology can be. And then, fasten your seatbelt because it's a ride. So thank you.
Hi, extremely impressive work. The way you've been working as a professor and also starting up many companies. I'm a biotech entrepreneur as well. And I was wondering, how do you manage your personal life with all this? And if you can tell us some tricks and tips. Thanks.
Sure. So that's not the question I expected today. So but I'm happy to answer it. So I'm a married mother of two. I live actually near my parents. I moved back to Boston to be near them so that they could know my kids and they could all grow up knowing their grandparents. And I'm pretty compartmentalized and really disciplined about my time, so I stay home on Wednesdays. I call that Mommy Day. I have date night with my husband every Friday night. I limit my travel. I actually think that's really important because you could spend 200 days a year on the road, and many of my colleagues do. I have a supportive partner. And I think everything else is just multitasking.
Hi, I'm Jerónimo from Argentina. Wanted to ask you about the initial moments of the development of these different processes, of these different projects. When you had this idea or this moment in which you realized this could have a practical application, did you start developing what would be like the concept practices in your lab, or did you start applying to grants or different funding ways to start this, like set up another place and start working on that project in order to then validate if it works and start like amplifying it?
Yeah, that's a great question. So I think for every project, the story is a little bit different. I will say there's a couple constraints that we think a lot about. So one of them is conflict of interest. And so we never have students working in companies, inventing for the company on an idea that's relevant to their graduation date. And so a lot of the time that constrains, the students are actually very entrepreneurial, they're interested in starting companies, but you can't actually start the company until the day they graduate. And so there are things that are happening around the time of a student peaking that I would say are kind of in the gray area, because they're actually pretty applied. As a team, you're getting pretty smart about what you think the use case is going to be, but it's not a company yet. Once you start the company and you've got the IP in, then that entity can move it forward with grant funding that is dedicated. I should say once that happens, at least the way we do it at MIT, and I always have the sort of like front page of the New York Times rule for myself, right? So like I really try to follow the rules. If it turned out in the press, you never want to be sad about it. Once it's in the company, you don't work on that anymore in your lab. And that's actually really hard. Some of these projects are things that we've worked on for 10 or 15 years. And so when we made the human microlivers, we put that in the company, and the company was going to work on toxicity and metabolism. There was a bit of an identity crisis for those of us in the lab who still love the liver. And actually, it was a great opportunity. We spent 10 years then working on liver infections, hepatitis, malaria, Ebola, other things. But you have to actually be willing to let it go and let the company do then what it's going to do, because it's like you're having another child.
What you mentioned about in terms of the IP, securing the IP, who is that is securing that? Like the university, the researchers, the students, all of them?
Yeah, so it depends on the institution. At MIT, things that happen that the professor involved in, the institution owns. And actually, you cannot be on both sides of that negotiation. So when you start a company, someone else in the company negotiates with your TLO office to get your IP into the company. And new inventions in the company may or may not involve you. Typically, they will not.
Hi, Sangeeta. Kara, I'm a PhD student here at Harvard Medical School. So I'm coming from a neuroscience background, and there's lots of discussion about organoids as models and how variable they can be and how difficult it is actually to test at least some basic science or even drug testing on them. So I was wondering if it was really exciting to hear about the liver and the organoids that you have there. I was wondering if there's, any considerations about similar the way, we have in the brain. Like, it's definitely a different system, but are they also variable, or how can we make them more as a good testing system in the case of your questions?
So there's a lot of questions in there. So so there are organoids in the liver. We are excited about them. I had dinner last night with Hans Cleavers. Some of you may know him. He's an organoid person. And I would say I think of organoids kind of like iPS cells, in the sense that I think the science is here, but the engineering isn't done yet. So they're not uniform, they're not scalable. There's lots of proof of concept in there. Can we definitively grow multicellular cell types from organoids today for clinical use? Not yet, but I think you could argue that it's the key science pieces, at least in some organ systems, have been tackled enough that you would want to scale it.
Great, thank you.
Mm-hmm.
So I just want to take a moment to thank all of our speakers in this series for really packing a lot of information into a short amount of time and sharing their wisdom with you. So if you just, for all the speakers, thank them too. So for the LOTs, you're now going to head to your assigned workshops, which are listed on the back of your badges. So the first workshop begins at 4:30, so we don't have that much time to get there because some of them are a little bit of a walk away. And so we have some volunteers who will be in the lobby to lead you there. And then after your first workshop, which runs for 15 minutes, you have 10 minutes to go to the next workshop. So it's really important that once the workshop is over, you move on to the next one. Don't linger at your first workshop. And then after your workshops are done, at 6:30, we're gonna have a reception at the Media Lab, and that address is also on your badge. So thank you.